So the last group of topics we're going to talk a little bit about organ transplants, and then we're going to get into some homeostatic imbalances. So some immunodeficiencies, so autoimmunity and hypersensitivities will be the last group of things that we're going to talk about. So when we talk about organ transplants, right, one of the key things about organ transplants is basically to provide an organ to an individual without rejection. Okay? So when we talk about organ transplants, there's 4 types or 4 varieties of transplants that occur. There are autografts. Auto means same. Okay? So in an autograft, you're basically taking a part or taking from one body site to another within the same person. Okay, so for example, you might transplant a toe onto a thumb, your big toe to become your thumb. Okay, or a skin graft would be an autograft. Okay, isografts. Okay, actually, I'd have to go back and look and see what auto means. Iso means same. Auto means, I have to go back and look at the prefix of auto. Isografts are between identical twins. Okay, and that is because they are genetically identical. We'll talk about why that becomes important here in the next slide. I think I talk about it a little bit. Yeah. Okay. Allografts. Okay. These are the most common types of grafts that when we think about an organ transplant, this is taking an organ or a structure from one person and transplanting it into another individual that is not an identical twin. And then xenografts are actually cross species. So taking a valve from a pig and transplanting it into a human. Okay. There's bigger structures there. I think there was a guy that lived for a certain number of months with an actual entire, I think it was a pig heart. Okay. While he waited for transplant. I think he ended up dying anyways, but you can transplant across species. Okay. And that would be known as a xenograft. Okay. So when you are testing somebody for compatibility with transplants, what they're examining is that MHC complex. Okay. Now again, we have thousands of MHC molecules on our cells. So what they're trying to do is match as closely as possible the MHC and the self-antigens amongst individuals. Okay. So that hopefully your immune system doesn't see that organ as foreign. Okay. So a transplant occurs, and after surgery, what they do is they suppress the immune system. So they put a patient on immunosuppressive therapy. And that's because, again, we don't want the immune system to recognize that graft, that transplant that has just been put inside their body. So they'll give corticosteroids to suppress inflammation, anti-proliferatives to prevent that proliferation and differentiation we just talked about from occurring, and then further immunosuppressive drugs that kind of dampen the entire immune system. Now again, not ideal because these all have fairly significant side effects. So when a person's immune system is suppressed to the degree that it has to be suppressed, with these immunosuppressive therapies, is a common cold can be deadly, right? So you have a patient who's now immunocompromised, okay? You have a pathogen, a common cold virus, bacteria that enters the body, and now you have no immune system to actually fight that off. So a lot of people in their early if it's not rejecting, the body's not rejecting it, succumb to infections. This is why it's so important when your patient has a transplant to maintain themselves in a sterile environment. So you really have to balance the immunosuppressive nature of the drugs you're administering with kind of the long-term survival goal. Okay, so there's this balance so that you have enough of an immune system to protect you from everyday pathogens, but not so much that your immune system is going to attack the organ that you have just been given. So best circumstances, rejection after 10 years in 50% of patients, meaning that within 10 years, 50% of people who've had an organ transplant will have rejected that transplant. And when we talk about rejection, this means the immune system has recognized that organ as foreign and starts to attack it. So, some research that's being done to induce kind of tolerance of the immune system, or of the foreign organ, is basically establishing something called a chimeric immune system. So what they do in these patients is they take the recipient and they suppress the immune system in that recipient. So essentially they will do they'll radiate the bone marrow. Okay. And what that does is it essentially destroys the majority of the cells of the bone marrow. And then what they do is they take the patient who's going to be donating the organ, okay? They take their bone marrow and they put it into the recipient, okay? We're not talking about organ yet, just bone marrow. So what happens is that bone marrow basically takes in that individual. And so now what happens is the B and T cells being created in that bone marrow are partly donor, partly host. Okay. And so what you hope is that this combined immune system is enough to mask the new organ coming in, so you have enough of the donor population, but not so much of the donor population that it's attacking all the other organs of the body. Okay? Kind of cool. Okay. So our first kind of homeostatic imbalance we're going to talk about are immunodeficiencies. Okay? And two immunodeficiencies we're going to talk about are SIDS, severe combined immunodeficiency syndrome. This is a congenital disorder. Okay? And what happens in this condition is it's a genetic defect. Okay? And what happens is you have a enzyme called adenosine deaminase. Okay, and adenosine deaminase essentially is mutated. Okay, so there's a genetic mutation to it. And when it becomes mutated, it creates a metabolite that is lethal to T cells. Okay, so what happens is you can't make T cells properly, which means you don't really get your B cells activated, because remember, our T cells are needed for our B cells. Okay, if left untreated, this is fatal. So it'll kill you. So typically the best course of treatment is a bone marrow transplant. Okay. The other immunodeficiency I want to talk about is AIDS, acquired immune, not immune, deficiency syndrome. Okay. So AIDS is caused by a virus known as the human immunodeficiency virus. It's transmitted via bodily fluids such as blood and semen, vaginal secretions. And HIV is a virus that loves to attack our CD4 receptors. Okay? So remember, our CD4 is going to become that T helper cell. And so what it does is it binds that CD4 cell, okay, and it injects its viral RNA into that cell, okay, into that CD4 cell. And what it does is it uses the machinery of the CD4 cell. There's an enzyme in there called reverse transcriptase. And it takes that viral RNA and converts it back into a DNA. Okay? And we know that DNA directs proteins, right? Okay, because DNA becomes a messenger RNA, which becomes a protein. Okay? So virus injects RNA, hijacks machinery to make DNA, okay, and then says, okay, host, go ahead, use that DNA as a template. Okay, this is called a provirus. So that DNA copy, okay, essentially directs the host to make more viral RNA and proteins. And this is how that virus replicates or produces inside of a cell. Okay? Now, there's a asymptomatic period, okay, that can actually be up to 10 years where those virus particles are just replicating. They're just dividing. And they're making more virus and more virus. And they just kind of sit around in your lymph nodes. However, eventually what they start to do is they start to destroy your T helper cell. The cells they infect, right? Because if it's a CD4, remember that CD4 is going to become a T helper, okay? So when this happens, you depress the cell-mediated immunity, right? And we already know, because hopefully at this point have ingrained in your head, without T helper, you have no immune response, okay? So people who acquire or who have been infected by HIV, okay, end up dying from AIDS, typically due to infection. With that said, okay, HIV reverse transcriptase, okay, has a ton of errors in it, which is really good for the virus because it makes targeting that first step really hard, okay? Because it has a high mutation rate, it makes drugs resistance really, really frequently, okay? So this is why early in the discovery and treatment of people with AIDS, it was so hard because they would make a drug that targets that reverse transcriptase, and then the virus would just mutate, and then that drug was no longer effective against that enzyme. However, over time, they created what's known as a cocktail, an AIDS cocktail. Not an AIDS cocktail, that sounds horrible. A cocktail against AIDS, okay? And what it is a combination of drugs that targets multiple aspects of how the HIV replicates. So they use things called fusion inhibitors. This blocks HIV entry into the cell even to begin with. They treat with integrase inhibitors. This blocks the viral RNA integration into the host's DNA so that that RNA doesn't become a permanent fixture to that CD4 cell. It has reverse transcriptase and protease inhibitors that inhibits the actual replication of the virus itself. So you can see because it has all of these safeguards. There's also a new antiretroviral, not new, but fairly new in terms of the window of HIV and AIDS. There's an antiviral gel that women can administer to the vaginal area that reduces the risk of getting AIDS by about 50%. Just so you know, do you know what protects you about 100% from getting AIDS? A condom. Okay. That's the easiest way to protect yourself. Okay. Okay. On to our autoimmune diseases. Okay. So an autoimmune disease results when the immune system loses the ability to distinguish self from foreign. So it begins to attack self and generating an immune response to self. So this creates something called autoimmunity, where the production of autoantibodies and sensitizes our T cytotoxic cells that then end up destroying our body. So some examples of this, for example, multiple sclerosis. MS is a condition in which we see the myelin sheaths of our nerves start to be destroyed. They know it's an autoimmune condition where the immune system starts to basically break down those myelin sheaths. They don't know what triggers it, okay? But they do know it's an autoimmune response. Myasthenia gravis, okay? This is also a neuromuscular condition. It's characterized by different degrees of muscle weakness. Again, another autoimmune disease, okay? Graves' disease, a condition of the thyroid. And then we have lupus and glomerular nephritis, as well as RA or rheumatoid arthritis, which is also an autoimmune disease. So typically what happens in these autoimmune diseases is we have kind of weakly self-reactive lymphocytes that may be activated in various mechanisms. So, for example, we might have a foreign antigen that may resemble a self-antigen. Okay? So, for example, the foreign antigen enters our body, we elicit an immune response to it, we now have memory, and all of a sudden, we don't realize that we actually have a self-antigen that looks really similar to that foreign antigen, and now that immune response starts to cross-react with those self-antigens. Okay? Another way in which these lymphocytes can start attacking self is a new antigen may actually appear on the surface of a cell that hasn't been recognized before. So, for example, you could have a genetic mutation that slightly alters a self-antigen, so now it looks differently. Okay? Changes in self-antigens by haptens, right? Haptens are those incomplete antigens, right? You get an incomplete antigen that now binds to a self-antigen which by itself doesn't do anything because the T cells and B cells have gone through that maturation process, that education. However, you add now a hapten to it, and now it thinks it's foreign because it doesn't recognize that molecule. Trauma can also elicit novel self-antigens. Okay? So again, the body typically doesn't see self-antigens as something foreign, but anything that either, whether it's something like close proximity that looks very similar or the generation of a new, will elicit an autoimmune response. Okay, on to the last topic, which are our hypersensitivities. So hypersensitivities are our immune's response to perceived, otherwise harmless threats that cause tissue damage. Okay, so they're different types of hypersensitivities, and they're distinguished by their time course, okay, so whether they're immediate or delayed, and whether there are antibodies, which implies that this is a B cell, it's supposed to be a parentheses, or T cells are involved, okay? So immediate and subacute hypersensitivities are antibody-mediated hypersensitivities. So again, that means these are controlled by our B cells. And delayed hypersensitivities are controlled or triggered by our T cells. So let's look at a few of these hypersensitivities. Acute type 1 hypersensitivities or immediate hypersensitivities, these are your allergies. They begin, like the name implies, almost immediately after contact with the allergen. Okay? Now, the key thing here is an individual has become sensitized in an immediate hypersensitivity. So the initial contact is actually asymptomatic, but it sensitizes the person to respond the next time. I'm going to jump forward and just show you what I mean by this. Well, actually, I'll go through the process and then we'll go look at the video, go look at the image, okay? So what happens is the following. We have a protein that's secreted by our T helper cell. Okay. This protein triggers, and we're just calling this a protein, this is called interleukin 4. Okay. This stimulates our B cells to produce IgEs. IgEs bind to our mast cells and our basophils. This binding triggers the release of histamine, induces an inflammatory response. Okay. Later encounter with the same antigen or allergen triggers an allergic reaction. Okay, I'm going to come back to that slide here. So let me show you what we mean by it sensitizes. Okay, so here is our allergen. This right here is our allergen. So over here it says antigen. I'm just going to put allergen next to it. Okay, first encounter. This allergen enters the body. T helper cell causes interleukin 4 to be released. Interleukin 4 targets our plasma cells and causes our plasma cells to produce a large amount of IgEs against this allergen. Okay? Now, these IgEs go and they bind to our mast cells and our basophils. Okay? So two populations of cells. Mast cells are similar to basophils, however, they're in the tissues of our body. They're not circulating like a basophil is. And they just bind to those cells. Nothing really happens at that point. Okay? However, the second time you encounter that antigen or that allergen, you now have a cell that is going to be receptive to these allergens. The allergen binds to the mast cell. The mast cell releases a massive amount of histamine. Okay? And this histamine will do a number of things. This histamine will cause constriction of the bronchioles. Can't breathe. Causes your tongue to swell. Can't breathe. You get a sudden vasodilation and fluid loss from your bloodstream. So typically, because of this massive dumping into our interstitial fluid, you go into circulatory collapse or hypotensive shock. And this is why things like bee stings, peanut allergies can cause death. Okay? Because of these two phenomenons, all caused by this histamine release. Now, very, very simple counteraction, something that counteracts this process very effectively, epinephrine. Okay? So this is often referred to as an EpiPen. That's the pharmaceutical marketed ones. You can also, this was under a whole bunch of scrutiny over the last few years because of the price gouging with EpiPens. You can also get epinephrine as an injectable. So you can get a syringe plus epinephrine vial, same thing, and inject it as well. Okay? So again, these are your typical like anaphylactic shock allergens. Okay. Those are immediate hypersensitivities. We also have subacute hypersensitivities. These are caused by IgMs and IgGs. Immediate, caused by IgEs. Okay. And they're transferred via blood, plasma, or serum. They have a slow onset. This is why they're called subacute. With a fairly long duration, so 10 to 15 hours. An example of a subacute hypersensitivity is what we call cytotoxic type 2 reactions. Okay? And we've talked about a transfusion reaction, but essentially when you give someone a mismatched blood transfusion, a cytotoxic type 2 cell reaction or type 2 hypersensitivity is what you see in those patients. Essentially, the antibodies will bind to the antigen. Okay, that's foreign. Stimulate phagocytosis and complement-mediated lysis of that cellular antigen. So that is why if we have a patient, right, with an A blood type, right, and we give this to a patient who has, here's our little Susie, okay, and she has B blood, we know that she creates A antibodies, right? Here's our transfusion reaction. Binds that. We know now that the binding of this antibody antigen complex triggers agglutination, and agglutination from the previous video or 2 videos ago triggers or stimulates phagocytosis, and now we get destruction of the red blood cells. Oh my gosh, so cool. Not cool if you're the person, but understanding how it's all coming together, right? We also have immune complex type 3 hypersensitivities. Okay. These are antigens that are widely distributed in our body or blood. They form insoluble antigen antibody complexes, and what happens is these complexes then deposit themselves in our tissues. Okay. And when they deposit themselves in our tissues, they essentially elicit an immune response to that area within the body, the tissues within the body that those complexes have actually deposited in. Okay, so it ends up causing kind of localized inflammatory responses where you get cell death and that type of thing. So, for example, lupus is a perfect example of an immune complex hypersensitivity. Our last hypersensitivity are delayed hypersensitivities. This is the only hypersensitivity that we talk about that has to do with our T cells. It's a super slow onset, usually 1 to 3 days. The mechanism depends on the helper T cells. Essentially what happens is the cytokine macrophages and our cytotoxic T cells become activated. They start to cause damage. Allergic contact dermatitis, so for example, poison ivy, is a perfect example of a delayed hypersensitivity. Tuberculosis, in terms of not the TB itself, but the TB test they use, skin test where they basically put a little bit of a solution under your skin and you come back in a few days to see if you're reactive to it. That system utilizes a delayed hypersensitivity mechanism to test for the presence of that pathogen or inside your body. Okay, so that completes chapter 21. Make sure you email me with any questions.